So people with Down syndrome make up the largest segment of the population at genetic risk for Alzheimer's disease, but our next talk is going to focus on a rare autosomal dominant form of Alzheimer's disease, and the presentation is going to be by my colleague, Dr. Jessica Langbaum from the Banner Alzheimer's Institute in Phoenix. Jessica is a true leader of the field. She does so much that it's hard to encapsulate it in a brief introduction, but she directs the Alzheimer's Prevention Initiative, the Alzheimer's Prevention Registry, the GeneMatch program. She's the incoming director of the the Arizona Alzheimer's Disease Research Center, and she's played a pivotal role in the trials she's going to talk about today, which were really innovative, unique, amazing, and I'm sure challenging studies in the largest familial kindred of autosomal dominant Alzheimer's disease, a group in Colombia, South America. So I'm grateful to her for joining us today. Jessica?
Wow, thank you, and what an honor to be here. I think I was here a few years ago, and it's lovely to be back and to follow such a wonderful talk on Down syndrome. I think it's such a natural progression, so thank you all. So yeah, I'm Jessica Langbaum, and it's really a partnership with the Alzheimer's Prevention Initiative that I lead with my colleagues, Dr. Eric Ryman, Pierre Theriault, and now Bob Alexander at Banner Alzheimer's Institute. But really, this is a global collaboration. So I'm going to step back a little bit, and I know we just heard a little bit about Alzheimer's disease, but here is a picture of the first person, Auguste Dieter, who Dr. Alzheimer saw, and this was the first case of Alzheimer's disease. And when she first came to present, she was 51 years of age, but when you look at her here, you would think she's much older. And she was confused and forgetful and having hallucinations. And she died at age 56. And we've learned a lot from her brain that I'll get to in a little bit about some clues into what might cause Alzheimer's disease happening at such an early age. So what is hereditary or autosomal dominant Alzheimer's disease? I know I get this question quite a lot. You know, I too have a family history of Alzheimer's disease. My grandfather died of Alzheimer's disease. However, that's very different having a first-degree relative or having a grandparent or two, or an aunt or an uncle, perhaps. That's not the same as autosomal dominant Alzheimer's disease. And this is generation after generation after generation, where about 50% of the family members have Alzheimer's disease, typically at a very young age. And so this is a genetic form of the disease. It's caused by a genetic mutation. And if the parent has the genetic mutation, their biological children have a 50% chance of inheriting that mutation. So you can see here it's a 50-50 chance of whether the parent with the mutation passes that along to their child. So what are some of the genes that are implicated in autosomal dominant Alzheimer's disease? And we just heard a little bit about Down syndrome with APP on chromosome 21. There are 50 different mutations on that APP gene. And this slide is probably outdated. So I'll just say there's probably more from when I took it here. But there's two other genes, presenilin 1 and presenilin 2, on chromosomes 14 and 1, respectively. And just look at the number of mutations on those genes, 150 mutations on the presenilin 1 gene. I'm going to get back to Auguste Dieter here. We learned many, many years later, right? Not at the time of her death, but many, many years later, she had a mutation on the presenilin 1 gene, right? So this was something we found very early on. She presented clinically at a very young age, but we didn't know what was causing it. It was because she had a very rare form of Alzheimer's disease with a mutation on the presenilin 1 gene. So there are Alzheimer's disease causing genetic mutations on the presenilin 1, presenilin 2, and APP genes. But there's also protective mutations on the APP gene, and perhaps this was some of the things that we can get into, the Icelandic mutation, where people with this mutation are protected against Alzheimer's disease. So these are some clues not only into what might cause Alzheimer's disease, but how we might prevent it. And quite a lot of work went into this on the protective factors on the APP gene with the identification of BACE inhibitor drug treatments and whatnot. And I want to point out that these are different than susceptibility genes. And this is not the focus of my talk today, but for instance, this is trisomy 21, I put as a susceptibility because there are people who don't quite live, they don't develop it. And so I think whether it's a genetic mutation and causing versus a susceptibility, but the APOE gene, for instance, having 1 or 2 copies of the E4 variant of the APOE4 allele increases a person's risk for developing Alzheimer's disease later in life, but it doesn't mean with 100% certainty that you will develop Alzheimer's disease. Like it seems to be, and I'm going to put a little asterisk there about seeming to be, because we're going to get back to perhaps some recent clues that there might even be some protective things that might happen to protect against people who happen to carry a genetic mutation on the presenilin 1 or presenilin 2 or APP gene that might protect them from developing Alzheimer's disease. So hold that for just a second. So these are very rare. These genetic mutations are very rare. They account for less than 1% of all Alzheimer's cases worldwide. However, Colombia in South America, this country, happens to be home to the world's largest extended family kindred of people with a single genetic mutation on the presenilin 1 gene, and this is the E280A mutation. How did this happen? How did Colombia get to be the home to the world's largest kindred? Well, it turns out that through quite a lot of research and searching through church records, including baptismal records, marriage certificates, death certificates, whatnot, we've pieced together, not we, the team in Colombia has pieced together what we think is the identification of a founder effect of a Spanish conquistador coming over to Colombia in the 17th century, settling there. This person had a genetic mutation, the presenilin 1 E280A mutation, settled there, had a family. And once you've been to Colombia, you can start to understand it's very mountainous regions. So settling, people live there, marriages, they stay put, and we have a founder effect. So Dr. Francisco Lopera, who leads a wonderful, wonderful research team at the University of Antioquia, he and his team started to notice people were coming to him and having Alzheimer's disease or symptoms of Alzheimer's disease at very young ages. And for many years, they weren't sure if this was perhaps a curse, what was happening, what was striking their families for developing these behaviors and these confusions at very, very young ages. And again, it was through the work with the families. It's a very rural area, not all of it, but many of these people are still living in very rural areas of the country. Meeting with the families, and there's some pictures of Dr. Lopera. I can't work the clicker, but sitting at the table with his silver hair, meeting with the families, earning their trust, getting to take blood samples, eventually getting to take, having family members agree to donate their brain, that they started to first realize this is Alzheimer's disease, and he and his colleagues, Ken Kosick and others, identified that it was the presenilin 1 E280A mutation. But when all this work was going on, we still didn't know, we thought it was a lot of people, but how many individuals were living with this disease, or living with this genetic mutation. So it turns out we formed a partnership with Dr. Lopera many years ago, over 10 years ago. And at the time, I think he knew of about 1,200 or 2,000, it was under 2,000 members of the family he knew of. Now we're over, he and his team have helped to identify over 6,000 living members. So we have, at the last count, we were just nearing that 6,000 people in our registry in Colombia. Of those, almost 1,200 are carrying this genetic mutation. And it's been a remarkable effort to identify, to earn the trust, and to genetically test all of these individuals. Now, the community standard here is to not know their genetic mutation status. So this is all done in a way, I just want to be clear, that the individuals, the families in Columbia, at this time, almost nobody wants to know whether they carry the genetic mutation or not. And we've respected that. So when does, I mentioned earlier, we weren't quite sure. The team in Columbia has done quite a lot of work about following these people over time, starting in very young child, when they're in the youth, starting around age 7, 8, 9. They can come in and have some cognitive testing, some perhaps giving blood when they're in their teenage years, and being followed over time. What we've found is that these individuals start to develop mild cognitive impairment, what we think of as mild cognitive impairment, around age 44. There's early subtle cognitive changes that we can pick up beforehand, before age 44, but what we call mild cognitive impairment, age 44. And full-blown dementia due to Alzheimer's disease about 10 years later, 54 years of age. When we were working with the family, we knew quite a lot about their clinical presentation, and this mirrors the symptoms, the cognitive impairment symptoms that these individuals have, mirrors what we see in the more common form of Alzheimer's disease, sporadic Alzheimer's disease in later life. This isn't the case with all autosomal dominant Alzheimer's disease mutations, but in this particular kindred of individuals with the pre-SNL1 E280 mutation, their cognitive symptoms match. What we weren't sure yet is, when do those biological changes start to take place? Could they be born with very high levels of amyloid? Or when does it start? Does it happen at such a young age that it might be too late to intervene? When we started our partnership with Dr. Lopera, they did not have the brain imaging facilities in Columbia to be able to answer this question, to be able to do those special type of amyloid PET scans that were so common now. So again, you have to think back to 2010, which seems like so long ago, but when we first formed our partnership in 2008, 2007, the imaging capabilities down there weren't quite what we were able to do here in the United States with the amyloid PET tracers. So in order to answer that question, when does amyloid deposition begin in this kindred, we actually flew about 50 to 60 kindred members of various age groups in symptomatic states from Medellin to Phoenix, Arizona in waves to do the brain imaging. And this was a remarkable experience. I have pictures of, they came in about groups of 10 to 15 and their family members, and they spent about 6 days with us in Arizona, and they would have, they met with our family and community services team to learn some coping skills and how to navigate through the disease course. We had dinners with their families. My children, who were very young at the time, have fond memories of dancing with the Colombians and sitting around eating many meals with them. So when does this begin? So this is an age, so we started, the youngest participants who had the brain scans were around age 20, all the way up, and we start to see amyloid deposition begin around age 28. So while this is the pre-SNL1 E280A is an amyloid it produces more amyloid in the brain, it's not that they're born with amyloid, or at least their brain is able to clear it. Maybe I should say that differently. But it starts to deposit in the brain at age 28, and then we see, but buildup happens. It's noticeable buildup that starts to happen around 20 years before before Alzheimer's dementia begins. Oops, let's see, okay. So this gave us the confidence that perhaps we could do a prevention study in people with this very rare form of Alzheimer's disease. And why start with this very rare form of Alzheimer's disease? Well, it's actually like the perfect scientific experiment, if you will. We have people with a single genetic mutation So they're very, in many respects, homogeneous. Not completely homogeneous, but a much more homogeneous population than, let's say, you might get in the more sporadic form of Alzheimer's disease. At the time, we had some interesting treatments to start to test. We were at the verge of we had a lot of brain imaging biomarkers. Now we have even more. We worked very hard to think about, well, what would be how could we measure change? And a lot of work went into that. And we really thought that if we could test first in this very rare form of Alzheimer's disease, we might be able to then set up for success in the more preventing the more sporadic form of Alzheimer's disease that occurs at later ages. So we moved forward. It was a very hard time to start studies. Back then, the NIH really didn't have a lot of money, and we kept moving forward. So we were very grateful that it was a top 10 world-changing idea by Scientific American. And here's the design of the study. It's really quite nice. We formed a partnership with Genentech and then Roche to study crenezumab, which is an anti-amyloid treatment thought to slow the production of amyloid in the brain. And we randomized mutation carriers to either treatment or placebo, and we had kindred non-carriers. So family members who did not have the mutation were included. They were assigned placebo. And this was all so that we can protect people from having to learn their genetic mutation status. Again, people did not want to know whether they carry the genetic mutation or not. So this was a study. We randomized the first participant in 2013, and we recently wrapped it up and completed database lock in April of 2012. So the earliest person enrolled in December 13 were followed until April of 2020, or sometime the longest person was on for about 8 years. Wow. People had to be on the study for a minimum of 5 years, so this was just screening what happened. Quite a lot of people volunteered. Some people weren't eligible, but we ended up randomizing 252 people. And this are their baseline characteristics. I realize it's very small, but they were generally well balanced, although you will see there were fewer women in the crenezumab carrier arm compared to the placebo arm. Something very interesting here that I'll just point out, or it's interesting to us as scientists, and that we'll be able to delve into more. Remember I said that amyloid deposition begins around age 28. People had to be between the ages of 30 and 60 to join this trial. A very, very large number of individuals in this study, much to our surprise, were amyloid negative. So they had no evidence that we could pick up on a PET scan of having fibrillar amyloid in their brains at the start of the trial. Just under half of the people were amyloid negative. So this was a remarkable study, an 8-year trial. So we had such high retention. People stayed in the study over 90%, well over 90%. It's remarkable. How did COVID impact? Well, that's that blue line going through this chart. And we had several dose changes. We learned a lot during the course of the study about what the optimal dose might be. But that blue line is when the country had to shut down due to the COVID pandemic. But very, very few doses were missed during COVID. It's really such remarkable adherence. So we had a few different outcomes of the study. I just put it up here. We had some cognitive outcomes, clinical outcomes, a lot of brain imaging and biological measures. Unfortunately, the treatment was not successful at slowing cognitive decline due to cognitive or clinical decline. And we had 2 different measures. But we've learned quite a lot. And I'm going to show you a few quick slides here. What's quite interesting, these were some of the primary outcomes, I apologize. But what you'll see here is that on the right favors crenezumab, on the left favors placebo. And although the treatment was not no outcome was statistically significant, all of the outcome measures moved a little bit in favor of crenezumab. And this was on all the primary and secondary outcomes. And here are some of the biomarker outcomes. So amyloid PET, tau PET, FDG PET, which is a measure of glucose metabolism, and some initial CSF measures. There is so much data, we're still analyzing it and more to come. Really, we just did our top line results presentation a few weeks ago. So where do we go from here? And I just want to leave us with some thoughts. Remember I said earlier there was a little asterisk about perhaps there might be something protective in some instances. And you might have seen this on the cover of the New York Times. It turns out there was a woman in her 70s who carried the Pre-1 E280A mutation, but she did not have any clinical symptoms. She had a head full of amyloid when we measured her with amyloid PET, but no clinical symptoms. And it turns out she had a rare protective mutation, a Christchurch mutation on the APOE4 that protected her from developing Alzheimer's disease Alzheimer's dementia, despite carrying this Pre-1 mutation. She did recently pass away, and so her brain and samples are going to really spur, I think, this next line of research. And then just a snapshot about where we're going with brain imaging and biomarker fluids. So measures in blood, cerebral spinal fluid, other biological measures. I think this is really how we're not only going to be able to do better clinical trials, but be able to go earlier and earlier and see if we can prevent this disease from happening. This takes a village. A trial like this, that was an 8-year study, a global partnership. There's even Dr. Frank LaFurla's name is on here, some UCI representation. This was people from Columbia, Genentech, Roche, Banner Alzheimer's Institute, advisors around the world. I can't thank them enough for allowing us to achieve this remarkable, remarkable study. And there's more to come. So I want to thank our funders, lots of support from the National Institutes on Health, pharmaceutical companies who partner with us on a number of studies, philanthropic support, and of course, our valued research volunteers, without whom none of this would be possible. So I want to thank you for your time today, and I hope I left you with a little bit of hope. Thank you.
Jessica, you and your colleagues are to be commended, congratulated. It's quite a feat. We have time for questions. See some hands.
Hi, thank you. Hello. Yeah, thank you. It's a wonderful presentation. By the way, I'm not a scientist. I don't really specialize in medical field at all, so I can ask a dumb question. I think your presentation, all the data we have seen focus on the treatment, effectiveness of treatment. So my question really is, when you talk about the gene mutation, if the family member has the gene with a potential Alzheimer mutation, 50% chance they pass to their children. So I think it's both good and bad. The percentage looks large, but on the other hand, there is a significant chance their children don't really experience this horrible mutation. So I do want to ask, based on your knowledge of the literature, what are the external factors which can contribute to the prevention of those kind of mutation? I heard, anecdotally, without checking, without validating, people say drinking coffee helps. People say exercising helps. So I feel interested. You perhaps read many, many articles. You can tell me clinically proved behavior adjustment as a normal person we can maybe adopt. Thank you.
Thanks for that question. So the lifestyle interventions are really, really important. I think we saw some of this even in the Down syndrome. So things like social engagement, exercise, especially exercise that increases your heart rate, staying engaged socially, mentally, all of those things are very, very important. We don't yet know if that will, what those interventions do for delaying the pathology of Alzheimer's disease. So it might be that our brains become a little bit more resilient to dealing with the same level of pathology in the brain, therefore staving off or delaying the onset of those clinical symptoms. So again, even in people with autosomal dominant Alzheimer's disease, with that genetic mutation, they still develop the dementia due to Alzheimer's disease. But not enough has been done on the lifestyle interventions to know how many years might we delay the onset of the clinical symptoms that appear in people with the disease or with the mutations.
Hi, I wondered, in your study, were you able to discern why so many participants did not want to know if they had the gene?
Oh, well, this is cultural. So And you'll see this when you go to Columbia. These are very, very close, tight-knit family, generations after generations, together, extended families. And you can imagine, you see this in other diseases too, if I learn, what does this mean for my brother, my sister, my aunt, uncle? It's a family decision. I think this will change. There's growing interest, but I think without an effective treatment to say that I could take if I was a mutation carrier, why no? Perhaps. Some people are deciding, well, maybe it might help inform whether I have a child or not. Others say this is God's will, right? So again, this is very, very complicated. It's not as much of a personal decision as a family decision.
Anyone else? Jessica, is it correct that people are still in the study?
Thank you for that. Yes, sorry, I blew over something. So although the main part of the study did conclude in a few months ago, people are still in the study. All of the carriers, in a blinded way, were moved over to active treatment. So if the carriers were on placebo, they are now receiving crenezumab. Again, blinded so that they don't have to know their genetic mutation status. The non-carriers are still receiving placebo. They'll be in this for about a year while we continue to analyze data and inform on next steps.
Great. You're up. Time for a couple more.
If a Spanish conquistador brought the genetic mutation from Spain, does Spain have a high incidence of Alzheimer's disease?
That's a great question. So I don't think Spain is different than any other country where there are genetic mutations present. I think Colombia became very unique because it's so mountainous and people settled in a way that didn't allow them, that they weren't leaving and spreading. So Spain, much different, people are moving around, so that mutation is probably around the globe. Colombia just happens to be a cluster of them.
Hi, thank you. It's really interesting to learn about the other genes and the genetic factors. My question is actually related to the APOE4 mutation. You mentioned that the likelihood is about a 50-50 chance of passing it on to your children. What do we know about someone who might carry a double copy of the APOE4 gene? Does that increase the likelihood that the gene is passed down?
So if you have 2 copies of APOE4, that means your biological child will have at least 1 copy. Because you are only able to pass down the E4 form of the gene. So APOE4, it's about people with 2 copies of APOE4 are about 3% of the general population. Risk of Alzheimer's disease, increases a little. Certainly, around about 30 to 50% chance of developing Alzheimer's disease in later life. Their age of onset is a little bit younger than people with 1 copy or no copies of the APOE4 gene.
Okay, one more.
Suppose a father has Alzheimer's and it has run in the family, the sister had it too, and the grown sons don't care to have the test. What kind of encouragement can we give to those grown sons that it's not the same as it was, that there's more hope now than we had before? What can we say to those sons?
Well, I'm not sure if the question is about genetic testing and counseling or just lifestyle or hope, but I think there's hope. That the amount of research going on is really wonderful. And even if they don't want to know whether they carry a genetic risk factor for developing the disease, I encourage everyone as much as possible to incorporate some of the healthy lifestyle changes that we do know. Again, they might not prevent it entirely, but they might sort of stave off. And certainly what's heart healthy is brain healthy. So exercise, nutrition, social engagement, mental engagement, all of those things we can all incorporate into our lives. And it can do us perhaps a little bit of good, not only for protection against Alzheimer's disease, but also our general lifestyle and health.
Yeah, I think it's important just to clarify too that the families that this study included are rare. And for people who have a family history of Alzheimer's or another form of dementia, we don't have definitive tests that can be used before a person has symptoms to say whether they will or will not someday develop Alzheimer's disease or other forms of dementia in most cases. So, you know, as far as Jessica said it beautifully, it's a very personal decision. People may or may not want to learn their risk information, but the tests available to us to assess that risk are still relatively limited. I want to thank Jessica once again. Fantastic talk, amazing work.